right, so Chapter 5 deals with the opposite of what Chapter 4 dealt with. Chapter 4 looked at how to grow microorganisms. Chapter 5 looks at how to kill them. So this is what we call control of microbial growth. And if you read your chapter, it goes through a little bit of history as to how the idea of we need to control microorganisms came about, which had to be proven that it was important to control microbial growth. There was a time in history when surgeons will go from one operation to the next without even washing their hands. The idea of washing, surgical instruments, was not thought of, was not important, and it had to be proven that there was an advantage to using aseptic techniques, to washing hands, to washing instruments, and until that was proven, you could not convince physicians that it was important. To them, it was a waste of time to go through all that protocol when there was another patient next door that needed attention. So these had to be proven. And once physicians began changing their protocols and embraced the idea of sterile fields and hand washing alone, the rate of patients who died from surgery decreased dramatically, as you might imagine. So to us may seem like a given that you have to wash your hands in a clinical setting, that you have to observe sterile techniques and sterile fields, but at one point it wasn't a given. It was something that had to be proven. Okay, so obviously times have changed, and when it comes to control of microbial growth, there are two approaches. There's the chemical approach and the physical approach. So physical methods will be methods that use things like heat, radiation, filtration, versus chemical methods that use chemicals. Levels of control are important because you have to follow the level of control needed according to each occasion. The most rigorous method of control is sterilization, which is the maximum method of control, which requires specific techniques, which are oftentimes expensive. So if it's not needed, then there's no need to go through that such a high level when only perhaps disinfection or degerming may be enough. So you have to know the definition of sterilization. Sterilization is the removal of all forms of microbial life. When you say all forms of microbial life, instead of saying all microorganisms, now you are encompassing all forms of microbial life. That includes endospores, that includes the cysts of protozoans, that includes viruses. So that is a general term that embraces all forms of microbial life. If you say the removal of all bacteria, that's not a sterilization, unless you say bacteria and endospores and algae and fungi, and you start naming everything. Okay, so this is a good way to define sterilization. So that a sterile item or environment is free from all kinds of microbes, including endospores and viruses. Now, prions are a different story. So even though we have not sterilized specifically for prions, because prions are not generally something that we have to worry about, we ignore the fact that the sterilization technique may not kill prions. If you're worried about prions, then you have to change the sterilization techniques to something even more rigorous than your standard. So lower than sterilization is disinfection. Disinfection seeks to eliminate most pathogens. So there is still some viable microorganisms after disinfection. So things like endospore, for example, may not be eliminated with disinfection. When disinfection is carried out on living tissue, we call it antisepsis. Disinfectants are used on inanimate objects. Antiseptics are used on living tissue. Oftentimes, the same chemical can be used. A disinfectant could be used as an antiseptic, except that it would have to be diluted in order so as not to become toxic. Antiseptics can be used as disinfectants, but then they will be mild disinfectants. So antiseptics are essentially less toxic because they're going to be used in non-living tissue. So diluted non-toxic disinfectants can be used as antiseptics. And these same antiseptics are diluted non-toxic disinfectants, meaning they're mild disinfectants. Okay. Another level of control is pasteurization. Pasteurization is a level of control that seeks to reduce the number of microorganisms in a food substance. Pasteurization is carried out typically on foods. It also seeks to destroy, to kill specific pathogens. So depending on the foods, it could be something like Listeria monocytogenes or Mycobacterium bovis. And it has to accomplish goals 1 and 2 without changing the taste and the consistency of the product. So these are the 3 goals of pasteurization. Notice that pasteurization is you could even call it, it's not disinfection, because disinfection seeks to kill all pathogens. Here, you're going after specific pathogens. Disinfection is not geared to reducing the number of microorganisms. Here, you are geared to reduce the number of microorganisms. Another level of control is decontamination. And in here, we're reducing the number of pathogens. So this is less than disinfection to a level considered safe to handle. Sanitation is even less than decontamination, because now you're going to reduce the microbial population, both pathogenic and non, to satisfy a standard, a public health standard. So here, you're just reducing the number of microorganisms without worrying as to whether some pathogens may be left behind. Preserving refers to the laying of spoilage, and again, it's typically done with in perishable items like food items, oftentimes including things that are that inhibit the growth of microorganisms, not necessarily kills them. So preservation could be putting something in a refrigerator. That's not going to kill anything. For the most part, it's just going to slow down growth. Degerming is very specific. It refers to a mechanical removal of microorganisms. So this is going to decrease the number of microorganisms by mechanical removal. Hand washing is degerming. Now, if you hand wash using an antiseptic soap, now that becomes antisepsis. That now you're looking at a higher level than degerming. So know the levels of control, keep them straight. A germicide kills germs, bactericidal kills bacteria specifically. Bacteriostatic inhibits the growth of bacteria, doesn't necessarily kill it. Now, what level of control you need depends on what you're trying to accomplish and the situation. So the level of control in a daily life versus a hospital will be very, very different. Daily life, hand washing is the single most important step to achieve control. There's no adverse reactions to hand washing, and typically regular soap will do it. Hand washing using antibacterial soaps is discouraged, because what you are doing is you are removing the easy to kill organisms, and the harder ones to kill remain. So you're selecting for the tougher ones, and leaving and getting rid of the mild ones. So hand washing is essentially degerming. It's a mechanical removal. It removes some of the normal flora of the skin, but more importantly, removes the transient microbiota. Normal flora is unaffected. It's sometimes affected a little bit, and the reason why it says it's unaffected is because a lot of the normal flora lives in deep layers, in crevices of the skin. So largely the ones that will be removed will be the ones at the surface. The ones hidden in the crevices will remain. Okay, so that's in daily life. In a hospital, the goal is to minimize the microbial population. Notice that the goal is to minimize, not eradicate. The reason why eradicate is not a goal is because it is impossible to eradicate the microbial population in a hospital. The goal is to minimize it so as to prevent nosocomial infections. Nosocomial infections are hospital-acquired infections. A hospital is a perfect storm for an infection. You have susceptible hosts, you have pathogens. All you need is a mode of transmission between one and the other. So that's what control is geared towards, preventing this spread of pathogens or opportunistic pathogens to susceptible hosts. On top of that, a lot of the pathogens present in a hospital have developed antimicrobial resistance due to the high use of antibiotics. In a water treatment plant, the goal is to assure that the drinking water is free of pathogens. Typically, we use chlorination for that purpose. However, chlorination has been known to produce byproducts, which have been deemed bad for our health. In water treatment plants, one of the worries is that some organisms have become resistant to chemical disinfectants, to chlorination. Notably, Cryptosporidium parvum, which causes diarrhea, is one of the main worries. And once in a while, if somebody in a neighborhood comes with Cryptosporidium parvum diarrhea, we know for sure this came from the water. So the physicians, the clinicians will inform the water treatment plants. Water treatment plants need to start taking steps to try to get rid of the organism. So this is one, and by the way, not too long ago, there was a case of not Cryptosporidium parvum, but actually what is called the Entamoeba histolytica, which is a brain-eating amoeba. A child died from it here in Texas, and it was in the water. So the water treatment plant has been shut down essentially until they can get rid of the organism. So that's a real thing to these organisms that become resistant to chlorination. So don't drink tap water. All right. So selection of antimicrobial procedure. Okay, so what method is best? That depends on the circumstances. There's nothing ideal. All methods have their pros and cons. What you need to consider are factors like, what are you trying to kill? How many of them are you trying to kill? What's the population like? What are the environmental conditions? What is the risk of infection? What is the composition of the items that you're trying to treat? So composition of items, are they made of plastic? Are they made of metal? So these are some of the considerations. So the most critical consideration is the first one. What are you trying to kill? What kinds of organisms are you worried about? How many of them are you thinking that the object or the environment has? So it's the most critical consideration because you have to ask yourself, are you dealing with organisms that are resistant, hard to kill, or susceptible? The most resistant form of life are bacterial endospores. So when you make a list of what is hardest to kill to easiest to kill. Hardest to kill endospores of bacteria are at the top, only lower than prions. Notice that the next organism right below endospores is mycobacteria. Below mycobacteria, another resistant form, which are the cysts of protozoans, which are dormant protozoans. Then we have the gram-negative bacteria, fungi. By the way, mycobacteria is a gram-positive, but it's a special gram-positive. Now, viruses are at the bottom. Envelope viruses are harder to kill than naked viruses. Oops, sorry, without naked, this is naked. So this is naked virus. Most viruses that infect humans are not naked. A naked virus will be, for example, rhinovirus. That's a naked virus, which is the virus that causes a common cold. But again, most human viruses are not naked. Most of them are enveloped. Below naked viruses, we have gram-positive bacteria. And then below that, the lowest thing would be the envelope viruses. Most human viruses are enveloped. Okay, HIV, the SARS-CoV-2 virus, the COVID-19 virus, all these viruses are envelope viruses. Okay, so compared to all other forms of microbial life, these are actually the easiest ones to get rid of, which gives you an idea of how hard it is to get rid of the other ones. Now, another point is that gram-negative bacteria here, some gram-negatives are going to be harder to kill than others. So we're going to speak about an organism called Pseudomonas, which it is a gram-negative, and it is very, very hard to kill. So this is a general list. So bacterial endospores are the hardest things, followed by mycobacterium bacteria, with Mycobacterium tuberculosis being the one that we're most worried about, is the most pathogen of the Mycobacterium. Mycobacterium leprosy is also bad, but we're not worried about Mycobacterium. It's not as prevalent worldwide, when you look at the world population. Keep in mind, about 1/4 of the world population has been exposed or has harbored at one point Mycobacterium tuberculosis or is currently infected. So it is incredibly prevalent worldwide. There is millions of people, millions that die every year of tuberculosis. So it is a huge problem worldwide. So controlling the organism is very hard because of the waxy cell wall, to the point that any type of mechanism will point out whether the mechanism is tuberculocidal or not. So tuberculocidal would mean it kills Mycobacterium tuberculosis. Okay, so notice that as far as the OSHA bloodborne pathogens, it is geared towards killing HIV, which that's an envelope virus, HBV, hepatitis V virus, and other envelope viruses, tuberculocidal, which is the one that is harder to kill than the other ones. Protozoan cysts are next, which are dormant forms of protozoa. These are typically excreted in feces, so you will be worried about this kind of organism if you're dealing with fecal contaminated items. Cysts of protozoans are typically resistant to disinfectants. Public pools. Yeah, public pools. You will be worried about public pools. That will be one place. Yep. Didn't think about chlorine pool. Yeah, I hadn't thought about public pools, but yes, that would be one place that you would be definitely want to have it heavily chlorinated and with a good filter. Pseudomonas species, remember that the next one was gram-negatives, right here. Among the gram-negatives, the worst is Pseudomonas species. So this is a genus of bacteria that is notably very resistant to chemicals. Disinfectants, that's really any chemicals, antibiotics and disinfectants. So this is one of the worst of the gram-negatives. Naked viruses come next, which is kind of puzzling because the naked virus has the capsid. This is a protein, And we're used to thinking of proteins as being very sensitive to the environment. And it's true. Also, what this is telling us is that the proteins of viruses are extremely stable proteins. Okay, so that's the first consideration. Pseudomonas aeruginosa can eat through the eyeball in 3 days, ophthalmic infections in 3 days. I believe it. It is a pretty bad bacteria. And it's actually hard to treat, extremely hard to treat. So no, just public pools, I guess. All right, well, you can't live in that kind of fear your entire life, but you're right. Yes, Pseudomonas aeruginosa is pretty bad. And yeah, it is a bad organism. As a matter of fact, if this is one organism that if it infects a operating room, they have to shut down the operating room because there's no way of getting rid of it. If the infection is bad enough, there's nothing you can do. You pretty much have to shut it down. It kills patients. Yeah, it is pretty bad. And see that one, Pseudomonas aeruginosa, it is an opportunistic pathogen. Remember that the opportunistic pathogens are common in the environment. Pseudomonas aeruginosa is common in the environment. Any place where you have water and dirt, you have Pseudomonas. Okay, so dirty water is Pseudomonas. But again, opportunistic pathogen. You have to be immunocompromised, very old, very young. For the most part, most of us should be able to deal, to not get infected with Pseudomonas, even though we're dealing with dirt and dirty water in our life, here or there. Okay. The other consideration is the numbers. Large population will take more time to control. Okay. So that's something else to consider. Just like we talked about generation time and in chapter 4, killing time is also constant. So it's a constant in that the same fraction of the population dies every set amount of time. So if 10% of a population is killed in 1 minute, then you know that out of the 90% that remained, the next minute 10% is going to die. And out of the 90% that remain on the next minute, another 10% are going to die. So there is a constant fraction of the population that dies during a given time interval, and that is so that makes the number of organisms more important, because you know that the more organisms, the longer it's going to take to kill. Having said that, washing first reduces killing time, because washing first removes organisms, and now you're going to have a lower population. So anytime that's possible, wash first, disinfect second. So decimal reduction time is essentially a measure of how long will it take to kill a population of organisms. It gives you the time required to kill 90% of a population under a specific condition. So the conditions have to be specified. So you can say that, that in this example, I'm trying to see through a, yes, yeah, we're going to go through the RT problems. Yeah, we're going to do this right now. So I'm just trying to see if here it tells me the conditions, and I don't see that it tells me the conditions. The 20 minutes. Okay. Typically, the conditions have to be specified. So let's just specify conditions. So let's say in this example that at 50 degrees centigrade, a population of bacteria has a DRT, a decimal reduction time, of 20 minutes. What that means is that every 20 minutes at 50 degrees centigrade, 90% of the population is killed. So if, for example, you start with 10 to the 8th organisms, which that means 100 million organisms, and they're exposed to 50 degrees centigrade for 20 minutes, the DRT is 20 minutes. That means that at the end of 20 minutes, 90% of the population has been killed. So instead of having 100 million, Now you have 10 million left because 10% were killed, 90% are still alive. So 90% of 100 million is 10 million. So now you have 10 million. After the next 20 minutes, out of the 10 million organisms, 10% have been killed. 10% of 10 million is 1 million. Am I right? Yeah. So that now you have, yeah, there we go. Now you have 10 to the 6 left. Okay. After the next 20 minutes, %90 have been killed, so 10% are left. Now we have 10 to the 5 organisms. And this continues on until there are no organisms left. So the key is 90% are killed, which still gives you 10% left. So you have to calculate what's 10% of the number, and that tells you the number of organisms left. So if we start out with 100 million, which is in this example what we started out with, and the reason why they chose 90%, the time that it takes 90% to be killed, is because that means 10% are left. And it's very easy to calculate 10% of a number. All you do to calculate 10% of a number is eliminate the last digit. You have 10% of the number. So if you kill 90% and 10% are left, 10% of 100 million is 10 million. That's why those numbers were chosen, because they're easy to work with. During the next 20 minutes, as long as the temperature remains the same, as long as the condition remains the same, the next 90% are killed, 10% are left. Okay, what's 10% of 10 million? Get rid of the last number. And now you have 1 million, 10 to the 6. And that continues on in that manner. So it will take 1 20-minute, 2 20 minutes, 3, 4, 5, 6, 7, 8 20-minute portions to be left with one organism. Okay, so that's how it will take to pretty much get rid of everything. I think there's another problem here at the end, and I will go through that one too. Environmental conditions also influence how effectively an organism will be killed. Typically, low pH, high temperatures make a better environment for killing microorganisms. So anytime that is possible, clean first, disinfect second, and then, if at all possible, as you're disinfecting, increase temperature, decrease pH, if possible. And it doesn't have to be a tremendous increase in temperature. A slight increase in temperature makes a huge difference. So warm water is a lot better than cold water, if possible. So when you wash with warm water. That will get rid of more organisms. Also note that organic materials can interfere with control mechanisms. Organic materials are things like dirt, blood, sputum, feces, anything from a human body or from nature will be organic materials. So organic materials oftentimes do a couple of things. They may allow for places to microorganisms to hide. They may deactivate chemicals, protect microorganisms. So those are the environmental conditions that you have to be aware of. So most chemicals are affected at higher temperatures and lower pH. So household bleach, can kill a suspension of Mycobacterium tuberculosis at 55 degrees centigrade in half the time as in 50 degrees. So by just, yes, so after disinfection, raise the temperature and lower the pH, or while disinfecting, raise the temperature and lower the pH. So if you can use a warm disinfectant, that can make all the difference if possible. Or wash with warm water, for example. Yeah, and lower pH, that sometimes is a little bit trickier to do, but the disinfection part, I mean the temperature part's oftentimes easier. And notice that it doesn't have to be a dramatic increase. 55, 50 degrees, that's warm water. That's a slightly warm water. That is a water in which you could take a bath, and it will be a nice warm bath. So it's not hot water. This is nice warm water. You can decrease the pH, that will be even better. But if you remember that most microorganisms, 55, 50 degrees, that's getting too high for them. Most organisms, 45 degrees is as high as it goes, and anything higher than that, when I'm talking about talking about the ones that live in human bodies. Human body organisms that can colonize human bodies, like 37 degrees, 40 degrees, 45. You make it into 50, 55 degrees, that's too hot for them. Okay. Other consideration was what kind of item you're trying to control to treat. So the critical items are the ones that are going to come in contact with body tissues, like a scalpel. Those will need to be sterilized. Semi-critical instruments are going to contact mucous membranes, and that could be something like an endoscope. So technically, does it need to be sterilized? It should be disinfected with a non-toxic method, so essentially with an antiseptic, because it's not going to go into the tissue, it's just going to go over a mucous membrane. Non-critical instruments, like stethoscopes, just need to be disinfected. They probably do sterilize colonoscopes, but they could just disinfect them, and that will be that will be the same. Sterilization, the techniques are fairly inexpensive in a clinical setting, and it just ensures that you're not going to introduce anything. So even though technically it doesn't have to, they probably do it anyway. So I would be surprised if they're not sterilized. I can assure you they are. Yeah. So they're giving you a choice. You could either sterilize or just disinfect with a non-toxic method. So if you don't have the sterilization techniques, you can just disinfect with a strong non-toxic disinfectant. But most places will have some form of autoclave where they could sterilize. Okay. So when it comes to, again, the first consideration, yeah, go ahead, Veronica. You have a question?
I do. I'm listening to you with the disinfectants and the sterilization. And when we disinfect things and we're using like household chemicals like bleaches, I guess we're getting mixed messages because we're being told that those chemicals are also being toxic for our body. So it's like a catch-22.
Yeah.
You're using it to disinfect, but yet you're causing toxins to your body. So I'm kind of confused on that.
Well, there's nothing perfect. So you have to weigh the pros and the cons. And that's really what it boils down to. So when you're dealing with, so I guess the first question is, what is the setting? Are you talking about your house, or are you talking about a clinical setting? If you're talking about a clinical setting, then you could be worrying about potential serious pathogens, in which case the potential for infection outweighs the potential for toxicity. If you're talking about a home where you may have children, then you're more worried about toxicity, because hopefully you're not worried about serious pathogens in your home. So in that setting, typically hand washing, mild disinfectants, just mild antiseptics will do the trick.
And I guess the reason I asked that is because my mother-in-law, who years ago passed from cancer, they were saying because she was a manager of housekeeping, which they do a lot of cleaning in these
Mhm.
hotels with these chemicals,
Yep.
and because of the years of exposure to it,
Oh yeah.
so that's why I said you hear all of this and it's just a lot. You like, I don't know if I should or shouldn't.
Right. Exactly. Yeah. Again, in her case, did she have a choice? I mean, no, that was, kind of, she was in a setting in which you did. Now, perhaps, depending on the setting, you might want to explore non-toxic type of disinfectants. Um, but in some settings, you may have no choice. Maybe wearing some type of, I don't know, airing the area a little bit, trying not to breathe the chemicals, wearing gloves so they don't come in contact with your skin. Yes, definitely. At the same time, you may also want to look at, ways to keep yourself healthy because no, you're being stressed with toxic stuff. So, that means that you have to eat better, not smoke, not drink. So you have to try to counteract. Um, so there's nothing perfect. That is the problem. That is exactly what you have to evaluate. So yeah, it is a catch-22. Absolutely, is a catch-22. But definitely, at a home setting, I would not use anything harsh unless there's a reason for it. Like there's somebody that's very sick, that is immunocompromised. That's different. In a home setting, no. If everybody's fine, healthy, I would just use a mild something or non-toxic. So no problem. Somebody's just asking when you discussing the setting, why do hospitals keep the temperature so low? Zinc option of killing some microorganisms with higher temperatures when cleaning. Keeping the room's temperature low will eradicate some microorganisms, or is there some other reason? Um, yeah, I do. They did in that sense. When you're saying hot, we're talking about really hot. So to keep a room warm, that temperature would be what, 25 degrees centigrade, 30 degrees centigrade. That's prime temperature for the growth of microorganisms. When we're talking about, uh, yeah, that would not do anything. So now you have two choices. Either keep something at a room temperature 25, 30, even 37 is hot. I mean, I know you guys are not used to, but I'm trying to think 37 would easily be 90 something degrees Fahrenheit, 90, what, 95 degrees Fahrenheit, maybe close to 100. That's not comfortable in a room either. And that is prime temperature for the growth of microorganisms. A high temperature that will kill microorganisms, now we're talking 50, 55, which I don't have a conversion here as far as, but degrees Fahrenheit, that is maybe 100 and, gosh, it's over 110 degrees Fahrenheit, I think. Or maybe it's about 110. That's way too hot. You cannot keep things that hot. So now you go the other way, cool, because cool is going to slow down microbial growth. So because you cannot go the high temperature route, you want to go the low temperature route. So that's why everything is kept cold. So keeping cold is not going to eradicate microorganisms, but it is going to slow down their growth, which is the next best thing because you cannot keep rooms at 100 degrees centigrade. So yeah, that's why. Okay, so okay, so things to consider when you're, for example, in just a practical application, in a burn unit, you're worried about Pseudomonas aeruginosa because that organism would kill a patient with burns easily because it's an immunocompromised patient. So they don't allow flowers or plants in a burn unit because anytime you have water and dirt mixed together, you have Pseudomonas aeruginosa. So they they don't have those. So think about, are you dealing with an area where there is dirt and water mixed? Okay, then you could be worried about Pseudomonas aeruginosa. Are you working with instruments that have respiratory droplets? You worry about Mycobacterium tuberculosis. In the case of Mycobacterium, the numbers of Mycobacterium will probably, that you may encounter, will be low, probably. However, it's a very tough organism. So again, you're considering numbers, you're considering what are you trying to kill, what's your worry. Okay. Organic matter, yeah, organic matter will, like sputum, okay, now we worry about Mycobacterium tuberculosis. Dirty water, Pseudomonas aeruginosa. Okay, so this is a DRT exercise. The first minute, 90% are killed, so the DRT is 1 minute, and the number of microorganisms is 1 million. Then that means that in the first minute, 10% are left, so it takes 90% are killed, 10% are left, so with 100,000. So this is 10 to the 6th. I can tell you 6 minutes to kill, to be left with 1, just because the DRT is 1 minute. So another minute will leave you with 10,000. Another minute will leave you with 1,000, 100, 10, 1, and then 7 minutes if you want to completely eliminate everything. So you will need 7 minutes to be sure everything is gone. Okay, so now we're going to discuss actual methods of control. And we're going to start with physical methods, and heat is the most common method to control microorganisms. There are 2 forms of heat, moist heat and dry heat. Moist heat is by far better than dry heat because moisture, the steam penetrates, which is something that dry heat doesn't do. And if you doubt me, just ask yourself, would you put your hand on top, not even inside, but just on top of a pot of steaming water, boiling water? No, you wouldn't. You'll get burned. Steaming water, 220 degrees centigrade. Would you stick your hand, don't touch the heating elements, but stick your hand inside an oven that's at 220 degrees centigrade, and would you get burned? No, you wouldn't. You do that all the time. You stick your hand in a 350 degree oven to get something out, and you don't get burned. If that was boiling water and 350 degrees, did I say centigrade? I meant Fahrenheit. 350 degrees Fahrenheit, and that's way past boiling water. And you stick your hand in that oven without any worry, as long as you don't touch any surface, the air itself is not hot enough to burn you, whereas 220 degrees Fahrenheit of boiling water is the air, the steam is hot enough to burn you. So that's the difference. The steam penetrates, so it's definitely better, more effective. Okay, so boiling water, 100 degrees centigrade, is not an effective means of sterilization because it does not destroy endospores. It is, however, an effective means of disinfection. And actually, not just endospores, but there are some bacteria capable of surviving being boiled at 100 degrees for 30 minutes. So again, it's not effective at sterilization, but is very effective at disinfection. Pasteurization, again, now this one is a hard one because there are many different methods of pasteurization. The deal with pasteurization is that the goal is never, it is not to sterilize. That's not one of the goals of sterilization. So you can say, no, it is not a method of sterilization. Okay, fine. What is a method of? Well, it is geared to destroying the specific pathogens to the point that if the specific pathogens are not being destroyed, the method is going to have to be changed so that it accomplishes that goal. Increase the shelf life, which essentially means lowering the number of microorganisms across the board, and it has to do 1 and 2 without changing the flavor and the texture of the product. Okay, now the classical method of pasteurization was 63 degrees for 30 minutes. And actually nowadays you can buy products like milk in which says on the label that it has been sterilized at low temperatures, meaning these 63 degrees for 30 minutes. Okay. The reason why is because the modern methods of pasteurization are incredibly harsh. HTST, high temperature short time, heats the product to 72 degrees centigrade for about 15 seconds, and then not only that, but then you have rapid cooling. So now you have high temperature for a short time and then rapid cooling. UHT, this is essentially a sterilization. This one heats something at 140 to 150 degrees centigrade, held for just seconds, and then again within the next 3-5 seconds it is cooled back down. That is a sterilization. So any type of product like juice or milk, cream that comes in those cardboard boxes that do not need refrigeration have been essentially sterilized. So they've been subject to UHT, and somewhere on that label it would say UHT. Okay, the milk that comes in the plastic jugs is HTST, unless you buy some brand that may say low temperature, with the idea that it's not a harsh method that completely destroys the food product. Whipping cream is always UHT? It probably is. Okay, interesting. Yeah, so it's ultra high pasteurized. And that's probably one of those cardboard things. And really what that means is that you don't have to refrigerate it. It's just that in this country, we're not used to seeing milk products out at room temperature, and that's why people refrigerate them. You don't have to refrigerate them until they're open, so you could easily leave them out in your pantry, and it wouldn't go bad. We don't do that because we're not used to it, but you probably could without any problem. That's essentially sterile. Okay, so that's pasteurization. Sterilization using an autoclave, meaning pressurized steam. This is the preferred method of sterilization. You do need a special machine. You need an autoclave, which is going to use steam at high pressure to sterilize. The parameters are 121 degrees centigrade, 15 pounds per square inch of pressure, 15 minutes. And yes, you do need to know the parameters. This kills everything, including endospores. Does not kill prions, at least not those parameters. You have to increase everything to kill prions. So 132, 15 psi, 4.5 hours will kill prions. Flash autoclaving for about 3 minutes, increase time, same pressure, and that will lower the time of sterilization. So essentially what you have is one chamber inside another. So this is 2 chambers. The inner chamber is where the objects are going to be placed. The outer chamber is going to contain the steam. So the steam will be pumped into this outer chamber and from there into the inner chamber, and is going to circulate and sterilize the items. In order for the item to be sterilized, the steam has to contact. Are all autoclaves pressurized steam, or is that a special type of autoclave? As far as I know, all autoclaves are pressurized steam. Otherwise, they're just an oven. If they're not pressurized steam, they're just an oven. Which you can sterilize with an oven. There's nothing wrong with that. It just takes longer. But an autoclave uses pressurized steam because that's what allows you to sterilize in less time. Because now you have the steam that can penetrate. The only problem with the autoclave is this right here. You're dealing with pressurized steam. And if you don't know what you're doing, accidents can happen. You can explode things, okay, which is not nice. So things that have to be considered with an autoclave, and one of the reasons why I have this here is because I assume some of you at one point or another in your professional careers, you may be called to deal with an autoclave. Maybe you work for a doctor's office and they have an autoclave there or something like that. So first thing is, can the item resist heat? This is something that cannot conduct heat or cannot resist heat. and it's something you want to use again and again, you may not want to autoclave it in this, sterilize it in this manner. Keep in mind that the steam has to contact the item for it to be sterilized, which means if it's in a bag, you have to open the bags, loose caps, place containers on their side. This also means that some things cannot be sterilized with an autoclave because the steam will not penetrate them. Typically, there's a temperature-sensitive tape that is put on the object, and that tells you that the correct temperature has been achieved. It does not tell you if the object is sterile. It just tells you the right temperature was reached. We don't know anything about the pressure or whether the object is sterile, but we do know the temperature was reached. Yes, it changes color. Actually, typically what it does is that these lines appear through it. So it looks like regular tape, and then after autoclaving, it's the regular tape with these gray lines all through it. That tells you it's reached the correct temperature. However, if you want to know for sure whether the autoclave is working properly and is sterilizing, then you need to use a biological indicator. So if you're dealing with an autoclave in a clinical setting, you have to keep records of having tested the autoclave every so often. So every 3 months or so, you have to run, or I don't know, honestly, I don't know how often, maybe it's every month, you have to run a biological indicator that assures you the autoclave is indeed sterilizing. The biological indicator are the heat-resistant endospores of an organism, of a bacteria called Bacillus staurothemophilus. If the autoclave destroys these endospores so that they're not viable anymore after autoclaving, then you can say, yes, the autoclave is definitely sterilizing. So that's the means to test the autoclave. Okay, wear safety equipment. And then something that is actually done, but is really questionable, is to wrap things in foil wrap because the foil may or may not prevent the steam from reaching objects. So you have to have an idea of what are the problems with an autoclave. A regular oven can also sterilize. It just needs more time and higher temperatures. So in what's called a regular oven, a dry oven, you can sterilize something by leaving it for about an hour and a half at 200 degrees centigrade. Whereas an autoclave will allow for lower temperature and less time. Okay, so also be able to know the parameters. Yes, the species of Bacillus staurothemophilus is heat resistant. So this is definitely a heat resistant endospore. So more so than other bacillus, and that's why that's the one that is used. Because if it kills that one, then it's going to kill all the other ones. Yeah, Staurothemophilus is a thermophile. Finally, another method of sterilizing using heat is incineration. The problem with this method is that you're burning something to ashes. So after you use it, you pretty much have destroyed the object. So it does have its place, but it is used to destroy, medical waste, animal carcasses that have been infected with some type of organism that has to be eradicated. This is what we do in a lab. When the inoculating loop is passed through the flame until it's red hot, we are essentially burning to ashes the organisms in the loop. Okay, so that's heat. Other physical methods involve filtration, irradiation, high pressure treatments. Okay, we'll talk a little bit about these. Filtration is, like the name implies, having a filter that has pores that are small enough to trap everything, including viruses, so that you can't take, you're going to have to use a liquid, you have to sterilize the liquid. So the liquid is passed through the filter, and the filter catches every single type of microorganism, and the liquid comes out of the other end sterile. This is very useful when you're dealing with liquids that are heat sensitive. It is also very expensive because these kinds of filters are very, very expensive. You can sterilize in this method with filtration liquids and air. Air can also be filtered and become sterile. Radiation, we have two types of radiation: gamma rays, which are ionizing radiation. These are the really, really strong forms of radiation. Non-ionizing radiation is UV light. We can use both to sterilize. Ionizing radiation is very, very damaging. It is what we call gamma rays. These are going to be your X-rays, for example. They are used to sterilize heat-sensitive materials, medications. The good thing about gamma rays is that they can go through objects. So you can have a medicine inside a bottle, and gamma rays are going to go through the bottle and sterilize the medicine inside. We use these gamma rays to irradiate fruits, vegetables, grains, meats. Spices are also radiated. I don't know if they still do it, but my understanding was at one point at least that if something had been irradiated with gamma rays, the package should say that. I don't know if it still does or not. Now, when you do it, some endospores are resistant to irradiation. However, those are not endospores that are medically important. Okay, so there are some bacteria out there that are resistant to ionizing radiation, but they're not bacteria that cause disease in humans. Non-ionizing radiation, UV light. These kinds of radiation, UV light, causes very specific damage in the DNA, causes something called thymine dimers, which we'll talk about in other chapters, in which two thymines get linked together and then the DNA is distorted. That creates mutations. So the reason why UV light can be used for sterilization when you're dealing with microorganisms is that microorganisms double in numbers so quickly that these kinds of mutations kill the organism very quickly. The only problem with UV light is that it doesn't go through objects. So it is useful to sterilize surfaces, like operating rooms, for example, may have a UV light. And when there's nobody in the room, they turn the UV light on, and it is sterilizing the surfaces. So that would be one use. We do use UV light to disinfect things like liquids, like water, for example. Also, it can damage eyes and skin. That's another problem with UV light. But as far as the sterilization, it can be used to sterilize surfaces. Okay, so that takes care of physical methods. Chemical methods. FDA is in charge of regulating antiseptics. EPA regulates disinfectants. What the chemical damages can be the cell membrane, proteins, DNA, depending on the chemical. Yeah, and when you're dealing with formulations, they typically combine more than one chemical. All right, so sterilants are going to be the highest, the most toxic chemicals. They can be used to sterilize some critical or semi-critical instruments. Lower than sterilants are going to be high-level disinfectants, which destroy viruses and vegetative cells. However, they're not going to destroy endospores, so they're not sterilants. Intermediate level disinfectants are going to kill live cells, fungi, most viruses. However, they're not going to kill Mycobacterium tuberculosis. They're not going to kill endospores, so they're not obviously not sterilants. Low-level disinfectants are good for fungi, live bacterial cells, envelope viruses, which are the easiest viruses to kill. However, again, cannot kill Mycobacterium, cannot kill naked viruses, cannot kill endospores. So the lowest level that kills viruses would be the intermediate level disinfectants. Oh, most viruses. Okay, fine. Yeah, I guess you're going to have to go to high-level disinfectant if you're worried about viruses, especially if you're worried about naked viruses. Things to consider: toxicity, activity of organic matter. This one is very important because oftentimes organic matter is going to deactivate the chemical. And in a clinical setting, you're worried about organic matter. You're worried about blood, sputum, feces, dirt itself, compatibility with the material being treated, if there's a residue, which because the residue could be toxic also, how much it costs, how available is it, how long does it last, can I store it, is it stable, and what do I do with it after I'm not using it anymore? Can I dump it under the sink, or do I need some type of a special procedure to get rid of it because it's toxic to the environment? Okay, so we're going to go through these disinfectants, or these chemicals, I should say, not necessarily disinfectants. There is a chart under study guides. I suggest you take a look at the chart, which summarizes this information. Okay, so alcohols. The thing about alcohols is obviously types of alcohols that we use to control microbial growth: isopropyl, which is the one you buy over the counter at the store. Ethyl alcohol is good too. The main thing about alcohols is the concentration. And the concentration range may vary slightly depending on the textbook. However, you may see it as low as 50%. You may see it as high as 90%. Yeah, the chart says, okay, 60 to 90%. Yeah, it may vary slightly. However, I would say the lowest range and the highest range, 50% at the lowest range, 92% at the highest range. Okay? So essentially, the main thing here is to know that a 40% concentration of alcohol is ineffective, because there's no chart that's going to say 40%. 50 is the lowest that you'll see it anywhere. Okay. You should know that 100% alcohol is bad too, because alcohols need water in order to act. So that's why 100% is not effective. If it's not mixed with water, it cannot affect living organisms or microbes. So that's essentially why. It needs to be mixed with water for it to work properly. It is not, in general, alcohols do not sterilize, they're not effective against endospores and some naked viruses. Another consideration, they do coagulate proteins and essential enzymes. So if you're working with an open wound, for example, where there is blood, the alcohol may coagulate those proteins. And the problem is sometimes microorganisms may hide in those coagulated proteins. It is commonly used as an antiseptic, sometimes used as a disinfectant, but it's mostly used as an antiseptic. Other limitations: evaporates quickly. Not going to, for example, on a surface, it's not going to be standing there for too long. May damage some materials such as rubber. Okay, so alcohols, putting down concentration is important. You should know what the limit, especially the low limit of concentration is, and the fact that it's typically better as an antiseptic than a disinfectant. Next group are very strong chemicals. These are the aldehydes. They are glutaraldehyde, formaldehyde. Nowadays, what we use is glutaraldehyde. 2% solution of glutaraldehyde is typically what is used. It is a sterilant, but you do need to submerge in the glutaraldehyde for a long time, for 10 to 12 hours to sterilize. 2 hours is enough for disinfection. Downside, it is toxic. So whatever is immersed into this solution will have to be rinsed. Okay. The other aldehyde is formaldehyde, which is extremely, extremely toxic. It is a carcinogen. and so it's not typically used. I mean, this is used for preserving specimens, for example. But even that, nowadays, glutaraldehyde is preferred. Okay, so the aldehydes are the first sterilants that we see. Bilguanides, like chlorhexidine, are very good antiseptics. They're good antiseptics because they have an affinity for mucous membranes, and they're very good against gram-positives, which are often found in mucous membranes. Have low toxicity. They're used in clinical settings in medical devices. A mouth rinse is a prescription mouth rinse, Peridex, which has a chlorhexidine. So not a sterilant, it's a very good antiseptic. Ethylene oxide, this is a gas, and it is used as a sterilant. You do need to use it in a special chamber, similar to an autoclave, except that it's using ethylene oxide instead of steam. It is used to sterilize heat-sensitive items, intricate items that are more difficult to sterilize with an autoclave. Problem, it is a mutagen, potential carcinogen. So whatever is sterilized using ethylene oxide will have to be aerated afterwards to get rid of the gas. Halogens, those will be your chlorine and iodine and other, like silver, for example, other metals. These are common disinfectants. Actually, iodine is an antiseptic. Chlorine is used as a disinfectant, especially of water. It is toxic, caustic to the skin, so obviously not being used as an antiseptic. Typically, a 1 to 10 to 1 to 100 dilution of bleach is pretty effective, except that, again, some protozoan like Cryptosporidium and Giardia are resistant to chlorination. Another problem with chlorine, it is inactivated by organic matter, and actually it's inactivated by light also. And that's why bleach is sold in a dark container, in an opaque container. Inactivated by organic matter, that is important. What that means is if dirt falls in a place where there's bleach, the dirt will inactivate the bleach. The bleach will be no good. So typically a solution of bleach doesn't last very long because it's just a matter of time. The minute you open it, stuff can fall in it. You might not even realize it, but it could. If the container is clear, light will be activated. So typically lasts for a couple of weeks, and then its potency begins to decline. Think about it. If you have a swimming pool, the minute a whole bunch of people swim in the swimming pool, the next day or within a matter of hours, the swimming pool turns green. And that's because organic matter, shed skin, dirt, that the people that swam was there in the pool, they released this organic matter that deactivated the chlorine, pool turns green. Same thing with leaf. When the leaves begin to fall on a swimming pool, the dirt deactivates the chlorine, pool turns green. So that's one problem with chlorine. Iodine is a very good antiseptic. It is not a sterilant. It doesn't reliably kill endospores, but it is a very good antiseptic. Nowadays, we don't use tincture, even though you could probably still get tincture in the store, because that's the one that is stains and it can be more of an irritant. And there are people that are allergic to iodine. So anybody who is allergic to shellfish is allergic to iodine. So they shouldn't be in contact with iodine. But what we do nowadays is we combine the iodine with an organic material into something called iodophore. Betadine is an iodophore. The idea is that the iodine will be released slowly. So it's not going to stain as much, and it's not going to be as much of an irritant. Again, very good disinfectant, antiseptic. Well, it could be a disinfectant also, except that it stains. One thing about it, though, it works as it dries. So when you use it, you put it on the skin, you let it dry, air dry. One downfall, there have been reports of Pseudomonas aeruginosa surviving stock solutions of iodine. So that's something to keep in mind. In general, metals are antibacterial. So things like silver have been used for that purpose for a long time. The 1% silver nitrate was used to prevent ophthalmic gonorrhea. Little drops in the eyes of newborn babies were used to prevent ophthalmic gonorrhea, so gonorrhea in the eyes, for many, many years. We have creams, antibacterial creams that contain silver. Zinc oxide are antifungal, so when you buy those little yellow antifungal medicines over the counter, they typically have zinc oxide. Desitin also has zinc oxide. Okay, so just to recap, sterilants that we have seen so far has been the aldehydes and ethylene oxide. Good antiseptics have been iodine and the biguanides, chlorhexidine. Strong disinfectants, aldehydes can be strong disinfectants. Mild disinfectants, or not as strong as aldehydes, alcohols. Chlorine is another disinfectant. All right. When it comes to Mycobacterium tuberculosis, anything that is sterilant obviously kills Mycobacterium tuberculosis. Peroxygens, these are there are two of those, hydrogen peroxide and peracetic acid. All right, they are good products. They're biodegradable. They're not toxic or very little toxicity. A lot less toxic than ethylene oxide or glutaraldehyde. However, how effective they are depends on what is being treated. When it comes to peracetic acid, which is the one that is most commonly used as a sterilant, it corrodes metal, so you cannot use it in things that are metal. All right, hydrogen peroxide, we use it as an antiseptic, even though it's really not a very good antiseptic. It is better, it's a better disinfectant than antiseptic. And the reason why is because catalases destroyed hydrogen peroxide. Okay, what are catalases? You should know that by now. That's chapter 4. These are the enzymes that destroy oxygen radicals. And many bacteria have catalases, notably Staphylococcus. Humans, we have catalases, which is all over our skin. So when you pour hydrogen peroxide on the skin, it begins to bubble. And that's because the catalases in the Staphylococcus that lives on the skin begins to destroy the hydrogen peroxide. If you pour hydrogen peroxide on an open wound with blood, it would also bubble because blood has catalases. So it is not a very effective antiseptic. It is more effective as a disinfectant. It is toxic too, so a little bit toxic. It leaves no residue, doesn't damage most materials. Hot solutions are used in the food industry. Vapor phase is used as a sterilant. You need a special machine to make it into the vapor phase, but here you have something that is fairly non-toxic that can be used as a sterilant. Peracetic acid is a very potent sterilant, very good because it is environmentally friendly. However, it is corrosive. It is an irritant. Okay. We're almost through. This is 2 more. Phenolics, these are very popular ones. These are your Lysol, your Anfil. You can buy these at the store. These are probably one of the first disinfectants ever used. As I've had Lysol, it's called Lysol. Yeah, it's a form of Lysol was the first thing that was ever used as a disinfectant. Okay, this is the deal with phenolics. This is important. These are the only chemicals that are not sterilants and are yet tuberculocidals at high concentrations. So this is the only product that even though they are not sterilants, they're still capable of killing Mycobacterium tuberculosis. It is not inactivated by organic matter like some of the other compounds. It is an irritant. It can be toxic. It does leave a residue. So we don't typically use it as an antiseptic, even though nowadays there are versions of phenolic compounds that are being used as antiseptic because we have make them mild enough for that. So physohex is a phenolic compound. Triclosan, hexachlorophene are, as a matter of fact, physohex is a hexachlorophene, are phenolics, mild phenolics. All right, finally, quaternary ammonium compounds. These are the mildest of disinfectants, which means they are antiseptics. They react with the cell membrane. They're non-toxic. They're used to disinfect food surfaces, food surfaces, I should say, surfaces where food is being prepared. They can also be used with soaps. So they can be used for washing and disinfecting at the same time. It is mild, so not effective against endospores, mycobacterium, naked viruses. Does destroy live bacteria, envelope viruses. This is a huge drawback. Pseudomonas aeruginosa can live on quads very, very easily. If you're trying to use quads on surfaces that are contaminated with dirty water, quads may not be the ideal thing to use. Scope, for example, has a quaternary ammonium compounds. Okay, that's it. Questions? So this is a little overwhelming, but it's just a matter of going back and maybe making lists of what's a sterilant, which ones are sterilant, which ones are disinfectants, which ones are antiseptic, which ones can be used in more than one way. Can be a sterilant and a disinfectant, and which ones are deactivated by organic matter. Yeah, that's basically what it boils down to. Any questions? So we can tackle chapter 6 next week. Yeah.
We swap lab and lecture night because Thursdays are when the test due date starts.
Okay.
And it feels like there's your lecture is really really important to the test taking. And so,
Okay.
like
Yeah, we can do that. Yes. But that would only give us 1 day to do the lab. Yeah, that would only give us 1 day to do the lab. However, at the same time, that if you've already started with the lab, we can ask questions about the lab. Yeah. So the next test for module 3,
it opens next Thursday, or the due date starts next Thursday.
the due date is next Thursday. Okay.
It starts that's the countdown.
Got it. Okay. So we really should deal with chapter 6. I'm thinking of doing it actually. Yeah, we're going to have to swap that because I have my 8-week classes ending. And so I'm kind of trying to wrap those up too. Okay. So yeah, we will go ahead and deal with chapter 6. And we can go week by week too. We don't have to do the same thing every week. So let's deal with chapter 6 early next week.
Okay.
And the last of the lab reports. Yeah, that's why I did not say Monday. That's why I said early next week because we're gonna swap. We're gonna do it on Tuesday, but I haven't set it up yet. So yeah, so we'll probably have to meet on Tuesday because there's something else going on Monday. So we'll meet on Tuesday to tackle chapter 6. The only good thing about chapter 6, to some extent, is that it is a review chapter. This is glycolysis, Krebs cycle, electron transport chain, which students should have seen these on Biology 1406. Now, we do look at it from the microbial point of view, but most of it is stuff that students have already seen. So we'll tackle it on Tuesday, and then on Thursday we'll do lab 6. Is that where we're at? Okay, staining, and then this week is 6. Okay, good. Which is the last of the labs before the practical. That is good. Okay, so let me look at the schedule. If I can move the lab for Wednesday, which I think I can, I'll do that. Because the way I have it right now, there's only one lecture that I have on Wednesday, so I could fit in 2. And that will give us a little bit more time for the lab. So let me do that. Let's meet on Tuesday for the lecture. And then on, okay. And then we'll meet on Wednesday for the lab. Okay.
Thanks so accommodating.
Oh no, that's absolutely no problem at all. Honestly, I mean, this is the purpose of this is to make it so that it's useful. Yeah. So we can try. Oh yeah, I apologize for this. Yeah, Mercedes. Yeah, lab report. I thought I had checked on it and I thought it was there. It's not. I'll go back and do that. Yeah, for some reason, some of the sections do not have their lab reports app. I'll go back and make sure that's right now. I'll take care of that right now. Okay. So yeah, definitely. All right. So we'll meet again early next week. And yeah, one more thing really quick. I did have an email from somebody who wanted me to go back and explain CFUs and dilution factors again. And I've been needing to find a time to do that. So what I may do, I may send an email out tonight, sometime tomorrow, but it would have to be during the day. If whoever can show up, whoever feels that this is something that they need, fine, they can show up and we can go over CFUs and dilution factors again. If you don't need it, if you're fine with it, don't worry about it. Okay. So we'll do it tomorrow, but it would probably be during the day. It would have to be maybe the afternoon, like 2 o'clock or something like that, which may not be the most convenient time for everybody. But anyway, yeah.
Will there be a review for the lab practical?
Yes, absolutely. Yeah, there will definitely be a review. And the week of the lab practical, so what I do is I open the lab practical and we have about a week to take it. I think that's how I have it in the schedule. So there will be a week in which there will be no lab reports. All there is the practical. So we can review it at the beginning of that week, and that way, and I'm going to open a study guide. It's just that I need lab report 6 before I open the study guide. We'll switch lecture and lab that week too. Yes, I would have no problem with lecture and lab that week too. Absolutely. Yeah. That's absolutely fine. You're talking about the week of the practical. Okay. So we can do that. That's no problem. So okay. So we'll see you again on Tuesday.
Okay.
Tomorrow. Okay. So have a good weekend. And